Actually, most of the time, I think one of the marvelous things about being in academic medicine, I think most of you know, is you get to travel and meet people. And I love the conversations because, as I was explaining to Kristen, who's a PA, as you know, when you put up your diagnosis of a young child, 3 years of age, her first thought in the U.S. was group A beta strep. That's what we see as the primary diagnosis. I shouldn't say primary, but as one of them. And when you listed 4 diseases or 5, none of which was group A beta strep, that's very good because we do exist in that globe, but have a very, and I love the TB because, again, we don't see a lot of TB in our hospital, and the biggest problem that we have is the follow-up. So because we are a more affluent society, one of the biggest issues has been contact management. And so when in one of the large clinics that I'm associated with is an HIV clinic, and tuberculosis isn't big on the list, but we do have people who actively babysit every day these patients who need to take antituberculous therapy, and that's a huge difference in the development of multi-drug resistance or other ones. So from my perspective, and as I mentioned, Kristen, I embarrassed you. I told them what your first diagnosis was of the dengue fever. Yes, and so, in her clinic, strep throat. Here in the rest of the world, it's something, which I think is the benefit of these programs. I mean, that's why I love to meet people here and go, because we look at it in our own isolated way, and then we have somebody from Brazil, and I told her she had snow in her country, and she goes, we don't even have any mountains with snow. So I'm learning geography as well as I'm learning microbiology. Listen, this is that lecture which was a little bit, I was surprised. The people at Omics asked me to give a unique lecture, and I wrote them 4 topics, and they chose this topic, which happens to be very interesting, but I was a little bit surprised that they asked me to do that, given what you've all done, which has been disease management. So what I'm going to do in the brief time that I have, an hour and a half, is that what you said? I have an hour and a half before lunch? Yeah, right. I want you to realize that the way I presented this was there's Louis Pasteur over there. Now, that's an older Louis, because Louis Pasteur actually has a lot of hair, or had a lot of hair. But I thought a good way to present this would be to kind of look at 3 time areas. What was the diagnostic lab in 1800? I'll be very brief because that's not particularly germane. What do you do in my lab today? And then I tried to predict, because we are looking very seriously at a revolution in microbiology. I think that it's not an evolution, it is a revolution. In my lab, in 3 years, will be so different than it was today when I go back and run the lab. We are making incredible strides. I, though, have the same opinion that was so beautifully presented by the woman who spoke, and I'm going to say her name wrong, our first speaker this morning, who pointed out we spend so much money on cancer and seem to have had such little impact. I love the new diagnostic procedures. I love MALDI-TOF. I love some of the Lyme molecular assays. My question is, will it make a difference in public health? Will the downstream consequences of the MALDI-TOF revolution see a better patient outcome? And I'm not sure of that answer. I'm really not. So I'm going to be very energetic about what I think I know we're going to do. There's no question the new molecular labs of 2025 will surely save money, but you know what? The laboratory doesn't see that money. It'll be in patient antimicrobials, downstream management, but the lab's going to be the same lab. We're always going to be yelled at because we spend money to make new tests. And with the Obamacare and the particular medical systems that I'm under, we are a cost center. And the only thing my administrator wants me to do is reduce staffing, increase the amount of time or decrease the amount of time it takes to make a test available, and do it with automation. And that's a very different world. I still like to pick up a plate, taste the colony. We were talking about that the other night. Look at it, smell it, get a good idea. But that's not where we're going to be in this very real future. So here we go. This is, I could spend hours on Louis Pasteur. We don't need to do that. This is a moot point amongst this group. But I have not given a lecture yet with Louis. And I've asked my lovely wife, who did take French and was a French major, and Kristen took French. I wanted to introduce this topic with a bunch of French phrases, and I was going to speak French to you, and I was turned down by my illustrious wife, so I guess I'll have to do that. No, I wasn't? I didn't. All right, so we'll move on. So look at this idea, and I thought the presentation of some of the diseases, the laboratories were created to solve disease problems. And when you look at a list, and I went through the WHO and looked up 1880, who was Louis last working with? I mean, the diseases were more of what you see than what I would see in my clinical lab. And so the laboratory was obviously based on what he was addressing, but I love the pictures of the original laboratory and the kind of environment, because when I started entering clinical micro in 1965, that was my lab. I mean, obviously there were benches, there were reagents, and Louis drove us with that principle, one bug, one disease concept. But we were growing microbes. And, when I looked at these pictures from his laboratory and looked at what kinds of things we were doing and who, in fact, was part of some of the original themes, I think of Louis Pasteur, I think of Robert Koch, who, by the way, if you read any history, did not like each other. They were always arguing, and one was trying to put the other one down, which sort of drove them both to be better. And then there was a gentleman in France named Bioméryeux, and why that's important, because some of you know the French systems and the Vitec, and Bioméryeux, who was early on, was a very strong partner with the secondary group of scientists in Louis Pasteur's laboratory. But the other person I sometimes think we forget, but you'll know the reagent. In fact, when I was having dinner last night, I was having dinner with a distant, how many relatives away, but the Ehrlich, Paul Ehrlich, was a huge laboratory principle of microbiology. You've all used the Ehrlich test. You've heard of dropping test and looking at zones in a medium. Was also an unrecognized, and I thought what you did was so classic. There are some very fine people in this era that we remember Louis Pasteur, we remember Robert Koch, we remember Ehrlich, but we forgot all the other people who along the way helped build a laboratory. And I don't want to spend a lot of time on those labs, but they taught us a very simple principle. It had to grow, you put it in the animal had to get the disease, and you had to recover a viable organism. Viability has been the driving influence of all of our clinical laboratories, which meant we were recovering a planktonic isolate. Good news, bad news. But the reality is that's what has driven me, and I've been a clinical microbiologist since 1969. One of the things I want to show you, though, which I really like, when I entered the clinical world of microbiology, now these are U.S. figures, but I think you'll get a kick out of this. Who was the president of the United States? We had Martin Luther King, obviously, who was doing a lot of things in our country. The value of what it cost us to live in this country. I remember the first new car my mom and dad had was about $3,000, and it was fun. Can you buy a car today that's a brand new car for $3,000? But importantly, how much did a blood agar plate cost? So when I started in microbiology, a blood agar plate cost 3 cents. What does a blood agar plate today cost us? Close to 40 cents. I mean, the consequence. So I love making this kind of slide, but the reality was, how many times have we taught our pathologist, the young lady of pathology, and we taught them so much about key reactions, and we talked about Kligler's ion agar, and we talked about K/A and biochemical reactions. We still do that, although we've a little bit amplified that scenario. So I love looking at the laboratory. I remember clearly the entero tube. Do all of you remember that narrow tube which had multiple media in it, and we pulled a line through it? It used these key reactions. Those reactions were originally defined by Louis Pasteur and Robert Koch. So although we were more mechanical, we still were using basically and have continued to use those things. And, our reactions were basically what I wanted to point here. What does it take us to identify a laboratory organism or significant isolate today? Generally, we're talking at least 24 to 48 hours. Now, the driving influence that I'm going to make a major point in a moment is in my diagnostic laboratory. So let me stop a minute, give you a little bit about that. We serve a fairly large hospital, about 800 beds in downtown Pittsburgh. I have 27 technologists in microbiology. We provide a 3-shift response. Our primary turnaround is a morning response and an afternoon response. But I'm asked by the administrators to make my lab match the shifts of the nursing staff. So they talk to us about shifts. So, you and I talk about hours. Administration says, what shift will you provide that result to? Again, a little bit off the line, but something that's very important to me is that the key people in my world are the infectious disease staff, the pharmacy staff, and what we used to call infection control, we now call in preventative infection management. Those people make my life worthwhile because they take the data that we have and make it have an impact downstream. But as far as administration is concerned, they want to know, can I turn a result around in a shift, 8 hours? That's the key. The other point that has changed dramatically is we have what we now call an intensivist, or remember, this word never existed when I started, a hospitalist. What the heck is a hospitalist? A hospitalist is an MD who is responsible for a particular unit, but in theory is there on a shift basis. So we have a hospitalist and each of these 8-hour shifts throughout the 24-hour day frame. That's very critical because with the bioinformatics that we now have, my staff can auto-release results that come out at 2 a.m. in the morning off our instruments. So if we have an auto-release of all the biochemical reactions at 2 a.m. and it goes to the floor because we can auto-release the results, who's going to act on that? Which physician is going to make a change in the antimicrobial therapy, which is usually the issue, or in managing sepsis, or cerebral spinal fluids, or mycobacterium? Because we can mechanically provide results every shift, 3 shifts a day, if somebody will act on it on the other end. So what is the most important comment here? Bioinformatics. How do we get the information from my lab through the hospital information system to the changing name of the person who is the hospitalist at 2 a.m. in the morning? And that has become a huge problem for us. But we have the ability, even with though we're doing things much more, and I'll go into this in just a little moment, much better than we did. The reality is, in my opinion, I'm still spending money. It's going to cost me $1.3 million to upgrade my methods, and I'm going to explain to you in a minute. I'll save the hospital money on the antibiotic usage, on the pharmacy impact of length of stay in antimicrobials, but none of that money is seen by me. I am still a cost center. So I have to drive the bus to get the better test results, but I'm still being responsible for the gas to drive the bus. That was pretty good. Gas to drive the bus. I'm still being, which is, I think, is pretty cool. All right, so this is a laboratory, you know, in the early 80s that again wasn't mine. I put this off the web. But you know, one of the things that we see is a lot of space. Fingerprints of equipment is an amazing issue because we're all being asked to do things with smaller space. So when you look at the laboratory space that we used to have, and we all had these islands, and we had people at each one of these benches walking around, we did actually put a device that could be measured by an actual light sensor in the ceiling. How many hours a day in your laboratory do techs walk back and forth and go from one instrument to another? That's a waste of time. These techs are not trained to carry a blood tube from one instrument to a Vitek to a microscope to another one. Why are we doing that? They build cars today in Detroit or around the world in which those instruments don't require but, what, 2 people that used to take 40? So one of the things I'm going to tell you is one of the benefits of automation in digital microbiology is that I will do the same test result that I'm now doing with 26 people. I'm going to administration and saying within 3 years I'll need 11 people. Because we can do so much better with automation, digital reading, and guess what? Robotics. So how do you transport specimens within your lab and plates? We now have a robot that does that. Can't you clean, haven't you seen on TV the robots that clean people's floors? My wife says, I want one. I said, no, you're the robot. But that didn't go over well at all. So that's why she's here and I'm still up here. But the bottom line is, why do we carry transport? Why do you take a tech that has an insurance policy, that has a vacation need to move specimens when you can do it with a robot? We have 3 robots in our lab that move blood samples from sample A to sample B. When they get tired, they don't go outside and need a break. They don't need food. They need a little electricity and some maintenance, which costs money. But the bottom line is robotics, robots, automation, and digital micro are here to stay. And we will cut our staffing by at least a third within the next 3 years. That's good news in a way. Why? Because in the U.S., the number of people in medical technology going into laboratory science is going down each year. If you look at the age of the MedTech society, it's almost 54 to the age of 54. Those people are not going to stay in the lab. So the laboratory that I'm going to show you in a moment, I get excited about this, is going to be quite a bit different. So let's not spend a lot of time on this. You know what the kind of lab, the space, the kinds of medium that we have used, and you know where we've been. But right now, the evolution turning to a revolution is based on this kind of technology, and I hate to talk about this. Do you know why? Mass spec is a chemistry analyzer. It measures either protein, DNA, or some other product. I hate chemists. I'm a microbiologist. Chemistry has always been a science that, you know, in the clinical laboratory, that's a chemistry procedure, that's a hematology procedure, it is a blood bank procedure, it is not a microbiology procedure. Whereas the only laboratory part that we don't allow people to enter in our database, that is, when the specimens come into the laboratory, we have to define a sputum. Did it come from an endotrach? Is it subepiglottic suction? What kind of specimen is this? Blood going to chemistry, blood going to hematology, anybody can enter it. We have to enter our own results because we don't trust how they would enter into the lab. So we're a different group. But you know what, ladies and gentlemen? We're not going to be different much longer. This is going to take us to a chemistry kind of environment where we are going to move plates by trays. We're going to have conveyor belts moving plates to incubators, and we're going to have robots carrying the specimens, and mass spec is going to give me a result in about 15 seconds. It's a whole revolution in microbiology. But let's talk just a little bit about where we are right now. My lab has just purchased a mass spec. There are two of them on the market. Sumatsu is out of Japan, which is a bioMérieux person. And the most famous, which you probably have seen or heard, is Bruker. It's a German system out of Germany. Hello. And it's been around since about 2007 because when Penny and I were on sabbatical in Cardiff, labs were beginning to buy the Bruker mass spec. Didn't have a large database, but now Sumatsu and Bruker are about the same. But let me just qualify that so if you ever have the thoughts, each of them can do about 200 organisms. Each of them has a small slide which you can run about 45 samples divided into thirds approximately, and you can enter that in and you can get a result once your system is running in about an hour of 45 samples. Now that's interesting because you've got a plate that's growing, so you're working on stools, he's working on urines, she's working on wounds. How do you fill up a little tray with 45 slots? Do you have it just, you have a mass spec, she has a mass spec, and he has a mass spec? So the strategy of movement of people, ergonics, becomes very important. So we're centrally locating a mass spec so that people don't have to move because the robot's going to take the slide from your station to his station to her station, and then one person will put it into the mass spec. I've lost a tech already. We haven't, but I have one less tech I need to carry things. You're going to be sitting there working on a continual sample base that's coming in, because what are you, stools? No, you're bloods or whatever. You're urines and whatever. So the whole setup of the microlab, because of mass spec. Now, those of you who may not know a lot about mass spec, and I don't want, because of my time constraint and lunch, the reality is we basically put on a very small, you know what microbe we need from a plate? I need enough to cover a toothpick. That's the inoculator. Now, please understand there's something bad about this that I don't like about MALDI-TOF. What is it? It's got to grow. You've got to have a plate. So that means the front end of our lab, before you saw the sample, and you saw the sample, sir, and she saw the sample, somebody has to set it up. Why do we have tech streaking plates? We build a car. Why do we have that? So today there are two very good systems. One's called Keystra and one's called Copath, which you can inoculate 5 plates simultaneously. You set the parameters, and they streak the plates very interestingly by having an inoculator put a drop on our seed of this plate. Then there's a magnetic bead underneath it, and you can determine the pattern you want the bead to roll on the plate, so you can isolate the organisms, and it's been proven. I'm a very good streaker. I've been streaking plates for a long, long time. I think I'm the best streaker in the world. But they've proven that if you use a magnetic bead that's being driven by a magnet below, and you separate on the same blood agar they will recover about 20% of organisms that I won't find in my manual streaking. It's true, it's proven. So why do we continue to manually streak before he gets and she gets their plate? So we are purchasing the Keystra, which will set up 5 plates simultaneously, and they will do it's not perfect for all specimens. We'll still need somebody working there, but my point is, I've just now reduced another tech, because I don't have the need for that tech to be streaking plates, which, by the way, is a huge waste of a very talented person's mind strength. Have you ever streaked 300 bloods that we get an hour? I mean, it's just, I love to walk by and say, so what are you thinking about? Because, they put a blood sample on there. What are you doing when you're streaking for bloods? I asked the tech, the tech's working in the hood. What are you thinking? It scares the heck out of them. So the point is, we put a very small specimen onto a plate. The plate, now here's the key, is incubated 4 hours. It's all I need. Where did this overnight growth come from, Louis Pasteur? I don't need overnight growth. I just need a little tiny colony. Your eyes aren't good enough. That's why we're going to digital imaging of the plates. We're going to use plates with chrome agar, which will give the small colony a very different shape and size, not detectable by me, but I'm going to illuminate that plate with some form of digital illumination. You'll see it in a moment. That plate will then be picked, and it will be then sent to you, because you have a workstation. You go and see the plates, not going there because of an image, because in the incubator is a TV camera. It looks at every plate, and then you will select which plates at 4 hours you want. Now, what did I just say? 4 hours. So he can be working twice. That's within a shift. So those plates will be coming to you. You'll be working continuously from those cultures that were set up so that the time you will see them now is going to be at 4 hours. 4 hours from 8 o'clock is 12. 4 hours from 10 o'clock is 2. You're going to be continually working with a 4-hour culture, placing onto a small site, and then we're going to run mass spec on it. I will have an answer back to the physician before he leaves the unit or she leaves the unit in the ICU. They're going to call up and going to say, that can't be my patient results because I'm still in the unit. I'm going to say, you asked me for a result in 8 hours, I just got it to you. Now the question is, will they work on it? It's great during the day. I don't know what it's going to be like at 2 a.m. in the morning. So going again back to the MALDI-TOF, the basic thought is we have a very small sample in a 45, it's about the size of a slide if you haven't seen it. It's about the size of a glass slide. It's divided into various sections, and Sumatsu and Brooker are a little different, and you can go through them, but basically they're the same. The reality is then we take, essentially in a vacuum, we make a very, it's like taking a very small sledgehammer. We beat the heck out of that microbial colony. But in doing so, we break apart its components. And we do so based on a molecular weight. And that molecular weight separates them when they move within this vacuum. So in essence, you are looking like a PGE, like a page, movement down a gel. But in this case, we're moving proteins that have been just absolutely destroyed in size and weight within a vacuum. And that vacuum is able to identify the length of time it took that protein to move within that field. And they, by molecular weight analysis, chemistry, begin to define what kind of proteins we have. So the other point I wanted to say about mass spec and the two systems, they both have about 200 isolates. Mycobacterium and fungi are part of that. Now, it's very limited in the mycobacteria. I don't think bovis, as we were talking about bovis, is there. But both of them have, now listen to the numbers, 5,000 additional species which are called research use only, RUO. So I've got 200 in either system, but I got 5,000 that are RUO, research. I can rerun that chip, which has those isolates, and if we're comfortable with sending those results out and have validated the other ones, I can find 5,000 results that match my 200. I can be sending out results to more organisms than you and I know. That's why I said to you yesterday, we're going to group them by phyla. Because we have so many names, nobody in the physician is going to understand. I don't know all those names. The physician's not going to know those names. So one of the benefits of the MALDI-TOF is to identify an organism that's viable. You had to grow it. but you can get many more identification parameters out of there. Now, both of them, without, again, getting lost, both of them have the capacity to do some genes. About 6 genes in either system can be picked up, which means today, if we're going through our lab of today, we still have to do susceptibility testing by standard methods. And if it's using a Vitek or something like that, that's still 6 to 8 more hours. So on the one hand, we can release an ID, but on the other hand, we can't give out a susceptibility. And what do most clinicians want? They don't care what I call it. They just want to know the susceptibility to it. So we've got to mix and match at the moment. So let me go through quickly what I really wanted you to see. And what MALDI-TOF does, in a very simplistic approach, is, you know what, it's really interesting? If you go to a car, you take your car through on the thruway and you throw in your money. You know, you got a dime, a quarter, or whatever. MALDI-TOF then takes like that machine and it just separates them all by molecular weight. So you just put a whole bunch of proteins and think of it as a whole bunch of coins, and then these coins are all lined up because these are the pennies, these are the dimes, these are the quarters, or whatever they are. And MALDI-TOF then takes this and you beat the heck out of that microbial colony. These proteins that are all weighed independently in a vacuum tube, which has a fingerprint. How big a vacuum tube are you going to take in your laboratory? How big is the base? That's a fingerprint. You must know that. And in essence, it says you have so many of these proteins that are this weight, and this weight. And then if you've had enough organisms that you've done this for thousands of times, you can match the profile. So it's a chemistry, I hate to say this, it's a chemistry analyzer being applied to microbiology, and that's okay. I mean, I'll learn to live with that, and I'll be happy for a while. So in essence, what you took is this, which is this whole bunch of coins. You separated them because you used laser separation and basically movement, and then you put them into a vacuum field, and you weighed them, and then the good part is you took all these and you weighed them according to an organism. So this is kind of what you get, and this is where you take your sample, 45 of them. The laser is here to essentially hybridize and to essentially make them come apart. And then this is moved in a field by weight, and then it's called time of flight. That's why it's called MALDI-TOF, T-O-F, time of flight within the vacuum. That's where it gets its name. So that's where we are. We have just purchased our MALDI-TOF, and it's wonderful, but again, we use the Vitek. And that's okay because in the laboratory today we can get the MALDI out, but we still have to do susceptibility testing. So let's look where I want to really go with this, and that's the new laboratory technologist. This is 20,025, right? These are the cone heads, right? You've all seen the cone heads on Saturday Night Live. Right now they've made a comeback in TV. But I was trying to make a joke about, I showed this to my staff. They were not happy with that particular picture of what the new technology will like. So first thing I want you to notice that we're going to have a tray which moves samples from place to place, and we're going to have robots that will be running samples from here. I want you to notice how clean that is. We will need about 3 techs to do what now we are running with about 10. Not saying all 26 are here in the laboratory at one time. The concept is that each station has a computer, and each station will be linked to an incubator in which there will be pictures taken continuously of the plates that are being incubated, because we can look at the colony and we will identify in 4 hours which plates can be pulled so that this gentleman, and that lady can identify those plates. Those plates will come out of an incubator, they'll go on a conveyor belt to each station where the tech never got up. You understand me? That's time wasted. People walking. It's not good for us. We want them to be productive all the time. So it's a continuous sample. So I think one of the things that's going to change in our lab, we still do all of our specimens by type. Stools, urines, bloods, respiratory. It's going to end. It's going to be now, but which specimen came in 4 hours ago? Which specimen came in 8 hours ago? We're just going to have a continual run. You're going to be working 8 hours, setting up samples. But here's the other point. Just think about this. All the side tests that you need for running a Staph X, the color reagents that we use, all the reagents that we do our complementary test off of a blood plate, you don't need them. No QC of those reagents. No storage. Your 4-degree refrigerator is going to hold Coke, Pepsi, and pizza. We don't need those reagents stored. It's going to be a very different environment. So if you look at that and then you look at this, I'm going to show you just 2 instruments that we're also using. I want you to see. I love this picture because I did not know that the Egyptians, now please help me, had many, many gods. I didn't realize the number of gods. But ibis, for which this instrument was identified, was, as I looked it up, now if I'm wrong, please take me into account that I'm an uneducated American about this, but in my database, it said Thor, number 9, was a god of wisdom, writing, and music. He was represented as a man with the head of an ibis, the bird. And this instrument that I'm going to show you right now is where we are also going, and it's where you will go. Not only will we have the MALDI-TOF, but this is called the IBIS T5000, and it combines the aspect that MALDI can't do. MALDI requires viability. Spinal fluids and blood cultures, I'm now being told by our infectious disease staff, we can't wait. If we have sepsis, one of you were showing slides this morning, if we have sepsis and the blood sample comes down to the lab, I want to know what it is now. And if we've got mycobacterium in the blood or spinal fluid, we can't wait for it to grow, even if we use expert systems. So one of the issues that has come with IBIS, in fact, the inventor of it received the Wall Street Journal, every year gives an award for a new instrumentation. The year the iPhone came out, this gentleman devised this system, and he won the award from the Wall Street Journal over the iPhone, which says a lot. But the IBIS system combines the thing that MALDI misses. It first amplifies the DNA or RNA of what specimens are there. Then it takes MALDI and separates the DNA, and then it gives us fluorescence at the end to identify visually what the organism is called. So please recognize that this is where we are going also. This is the IBIS. It's now owned by Abbott. And what it allows us to do is to amplify the DNA first, to then recognize after the 2 to 3 hour amplification that we can use MALDI on the DNA, and then to get a final answer by after the MALDI-TOF of having an answer for those organisms that don't grow. So the new lab, here's the word, I want you to remember this, is culture omics. Culture, we'll use MALDI up front because it grows. And we'll use that on urines, stools, sputums. We'll use the IBIS T5000 on spinal fluids and bloods because it doesn't require viability. That's why the new lab will be culture omics. We'll still do some traditional cultures, about 20%, but we'll almost run our entire lab by this method with MALDI because I don't want to spend the time growing the organism. I don't need to grow the organism. I can detect its DNA. The problem with that is, how many more organisms are we going to find? And what does it mean to the clinician? If I sent out a report from a stool where traditionally we're looking for salmonella or some other stool pathogen, I'll find what, 800 organisms? That's why phyla clustering is where we're also going to be. And I know my time is up, so I don't want to spend a lot of time with this, but I just want to show you why this gentleman who we had dinner with last night, Garth Ehrlich, decided he could combine 16 primers in an instrument that would cover every known clinically important microorganism and expand its DNA so it could target DNA in one run. So this is the kind of results, and each color is a different phyla. And each phyla has a specific kind of primer, and so that every organism, remember I talked about the Firmicutes, and I talked about, well, chlamydia for sure, every one of these organisms can be identified in the IBIS system within 4 hours. And I don't need it to grow in a plate. So this is culture omics. So the laboratory of 2025 will be running this instrument. And the only reason I want to show you this, one of the biggest problems we have at our hospital in Pittsburgh is replacements, biofilms, but particularly joints, weight bearing up. And what does the microbiology lab find? Nothing. Culture negative. And the surgeon comes down and says, and Nikita Khrushchev's kind of idea, takes a shoe off straps my desk and says, John, what is the heck wrong with you? I have these knee replacements that are always coming loose. We have to redo the procedure, and you telling me there's nothing growing there. I said, well, you know, I'm Louis Pasteur. We culture. These are from studies of knees in which the culture was negative. Look at the, you can't read them, but look at the number of organisms we can find using IBIS. Look at the number of organisms. Now the question is, doc, what are you going to do with that information? So the molecular driving revolution will make things very different. Last slide I just want to show you is, I have another I've been so fortunate to meet some outstanding scientists. The other person is someone who said, look, all of you do this sometimes. Get a plate. It won't work with mycobacteria, but if you grew staph on a plate, a blood auger, McConkey's, whatever, take a green pen, a laser, put it underneath it, and hold it against the wall. You'll see a pattern. Every microbe grows as a colony. And if you illuminate that pattern from underneath, you ought to be able to see different harmonics of wavelengths. So what he did is, and what we started to test for him, is we could separate all kinds of patterns. Here's the kind, it's really remarkable. A very small fingerprint, very small pattern. And you know what this does? Folks, I can now distinguish particularly those organisms that are, is it the same staph in your sputum that's in your blood culture? Because I can get the same pattern. The identification would all say Staph aureus. But is it the same Staph aureus that's in the blood, spinal fluid, and urine? We can't do that now. This kind of digital microbiology, coupled with MALDI, coupled with IBIS, will not only allow us to be better and faster, but I'm going to say to you, sir, the issue of your disease, blood, urine, spinal fluid, it's the same microbe. Or, unfortunately, ma'am, your blood culture is a Staph aureus, MRSA, if you will, but it's not the same MRSA that you've got on your hands, or something like that. So the new micro lab will be automated up front. It'll use robotics. It'll use the molecular detection of the omics part, and we will turn results out in our new lab within 2025 in surely 8 hours. And I'll do it with 1/3 the staff that I now have, which administration loves. That's what's going to buy me this new instrumentation. So I think I just wanted, these are some of the patterns that we did on wound isolates. They were all the same wound isolate, but we were able to separate the patterns. You know how long it gets a pattern? About 8 hours, about 20 seconds on each plate. It moves around, they measure the patterns, and so we couple that with IBIS, with MALDI-TOF. I can tell you an awful lot about the microbiology. So I don't want to show you these. Time is up. I just want to show you how we're integrating this into our system. So the last thing I want to say to you is, you know what this is going to charge you with a problem? How are you going to report this? You have a whole cadre of microbes. So the idea is that we will still now, this is my interpretation, but I think you'll like this. I hope it's as revolutionary as yours, because I already said to the audience, I thought what you said about cancer therapy, I sort of think about microbiology. I've been the bastard child of the laboratory science. People beat on me all the time because my results take so long to get to the patient's records. You know what? That's not going to be the problem in the years shortly coming. We're going to make the results available. Now they better act on them. So I point out to you, what I say now is the microbiology of the laboratory report will still do some culturing. We're not going to run MALDI and IBIS on an outpatient urine. I mean, come on, that's an E. coli. We can do a plate test. It's cheap. We are going to run the molecular aspect, but you know what else we're now going to have? This is where microbiology is going to make itself an immunologic cascade understanding. When I recover all of these organisms, I'm going to have a health signature of that patient's normal flora. So we're going to have a traditional culture, we're going to have the molecular and we're also going to have with a probable recognition of so many organisms, we're going to relate them to health of the patient. And I'm going to call that a health status. Two other things I might point out to you. Are we going to be doing biofilm staging? I think in certain wounds we will. Will we look at every patient who comes in? Are we going to, would you ever give a transplant today without knowing what the ABO group is? Of course not. Why do we give fecal transplant and know what the enterotype is? Why don't we do a rectal swab on every patient who's going to get a fecal transplant? We will be, because you know what? This methodology is going to tell me all about that person's stool. So one of the things I want you to think about, you got a patient with MTB, maybe we should do a stool culture to find out their status. Not on TB, but on their health microbial status. So I also say, because I'm associated with the dental school, when we start talking about transplants, as you know, there's an awful lot of now transplants in teeth. Maybe the first thing we ought to do with a new dental patient is have them bend over and get a rectal swab, so we can find out what their normal rectal flora is, because that might have an influence on what kind of transplant will be used. So things in microbiology, I hope you'll appreciate that, are going to change dramatically. That's the new microlab. We're not going to have an old classic microlab. It's going to essentially be a sterile environment where things are moved through automation. Hear me. Automation, robotics, digital micro. That's the Laboratory 2025. It'll use my clock because we'll make things a sort of understanding. And I hope Louis Pasteur's in heaven when I get hopefully a chance to meet him. And he probably won't be very happy. But Louis' era is coming to an end. We're going to look at multiple organisms and multiple diseases identified by systems he never had the option of knowing about. It is a whole new world in clinical microbiology. It's a revolution. Thank you very much.